You can do science. You can do also science as a 9-to-5 job, but then you're most likely not being competitive. Science for me means I cannot let go. The area that I'm working in is called structural biology. This means we approach science by visualization. Because, of course, proteins or biomolecules are not visible to the human eye, they are too small. But what we can use is experimental methods to make them visual and present them. And this is what our core expertise and core research area is, to make proteins, protein assemblies, molecular machines visible. That's why I think proteins are very beautiful sculptures. So I'm very interested in art, architecture and design. I like looking at these things. I like the Bauhaus. I like Mies van der Rohe's pavilions that have this super simplicity. I find aesthetics highly important. If you spend 2 or 3 years to get a snapshot of this picture and then you visualize it, of course it's important to visualize it in the most beautiful way. I never considered myself really a writer, so I really didn't like much writing essays at school. Now when we write publications, what we have to do is show our science in a kind of a story. I can use art and storytelling to visualize these discoveries that we have, to make people aware of what we found, to engage people and make it accessible to non-experts, to the public audience. Making discoveries is one of the most exciting things that you can imagine. You're sitting there and you spend years and years to get to this point. And finally you have achieved it. This is the moment where you run around the lab, your heart goes up and down. You say, everybody, come to my office, I have to show you something. It's amazing. We can see how this thing works now. One of the major goals of the next scientific breakthrough is, can we have synthetic cells? Can we reconstitute a cell from isolated individual components? So far we often have removed components from the cell and ask what happens to the cell. But ideally we would like to really create synthetic cells with all the components that can be manipulated and controlled. This is one of the major breakthroughs that I hope in the next 10-20 years will come.